Today we are going to study the structure of skeletal muscle. Okay, so let's start the topic. Firstly, we need to know the location of skeletal muscle. Skeletal muscle is connected to its respective bone, you can see, with the help of a specialized structure which is known as tendon. Tendon is connecting this skeletal muscle to its respective bone. Now, when we open up a little portion of the skeletal muscle, then you can see that the skeletal muscle, this is the little portion of the skeletal muscle, and it has the epimysium, the outer layer which is known as epimysium. Now, in the epimysium, there is another layer which is the perimysium, and perimysium is enclosing the muscle bundles in it. These red dots are showing you muscle bundles, and the perimysium is shown by the black round. So the epimysium and then the perimysium which is enclosing the muscle bundle. Okay, now one more thing, that is the epimysium, perimysium and endomysium. These terminologies are not the part of your A-level syllabus. This is just for your knowledge and understanding. So you can see the muscle bundles are present in this structure. Now we are just taking one muscle bundle from these muscle bundles. So this is one complete muscle bundle, and it has the endomysium, you can see, and it has further units, and those units are known as muscle fibers. You can see the red dots, and these red dots are representing you muscle fibers. Now this is muscle fiber. Out of these muscle fibers, we are taking only one muscle fiber to understand the structure. So this is muscle fiber, and muscle fiber is one complete skeletal muscle cell. And as we know, it is a striated muscle, so it has some light and dark regions on it, you can see, and it is syncytium, so that is why it is multinucleated. You can find out the three nucleus in this diagram. Now, in the muscle fiber, means if it's this is a one complete cell, then it has further units in its structure. So in the muscle fiber there are myofibrils. These red dots, you can see further on, these red dots are present here. So these red dots are representing you myofibrils. This is one myofibril. Out of these myofibrils, we are taking only one myofibrils. Now, the myofibril has furthermore contractile units, which are actually the functional units of this muscle fiber. So, the contractile unit, these are known as the sarcomere. Sarcomere, this region, you can see this is known as sarcomere. So, what is a sarcomere? Sarcomere has, you can see that the two types of regions, the dark region and the light region. So, two light regions and one dark region is shown in the sarcomere. And actually a sarcomere has two light bands on its edges and one dark band. So this is sarcomere. Now when we further study the structure of the parts of the sarcomere and its structure, then we can find out that the sarcomere has myofilaments. Myofilaments are actually the dark and light regions which are found on the structure of the muscle. Means on the sarcomere there are the two regions, the dark regions and light regions. So these dark and light regions are made of the two types of filaments. So those two filaments are making the myofilament. These two filaments are making the myofilament. So myofilament has the two types, that is the actin and myosin. What is actin? Actin is this light region. This light region is showing you actin, and actin is also known as the thin filament. Actin is also known as the thin filament, and this region is showing you the dark region, and this dark region is made of the myosin filament, and this is the thick filament. So the thick filament and thin filament, these are making the myofilament. So I'm revising it again. What is the order of a muscle? You can see this is the muscle, this is the little part of the muscle, and it has number of muscle bundles in it. Now inside the muscle bundle, one muscle bundle, there is a muscle fiber, and inside the muscle fiber there are myofibrils, and inside the myofibril, what is present? The sarcomere. And sarcomere has the two types of filaments. and together these two filaments are known as the myofilaments. Those two filaments are actin and myosin. Actin is the thin filament and myosin is the thick filament. So this is the structure of the muscle. But we have further some diagrams, and through those diagrams we can further more study the structure of the muscle. You can see this is the myofibril and this is the sarcomere. Sarcomere, you can see this is the myosin, that is the thick filament, and this is the actin, which is the thin filament, and the Z lines on both of its sides. The Z line is present. We can see later on what is the Z line. Okay, but firstly we need to study the structure of myofibril. What other things are present in a myofibril which we haven't seen in the previous topic? Okay, now you can see the myofibril has the two types of filaments, the thick filament and thin filament. So now firstly we are going to study the thin filament. This is the thin filament. You can see these are the two thin filaments and this is the thick filament, and these are actually making the myofibril. So this is the thin filament. Thin filament, you can see it has actin protein. What is actin? What is the structure of this actin? Actin has the two spiral strands. You can see these two spiral strands which are twisted around each other. So these are making the actin protein. So this complete structure is the actin. Now, along with this actin, there are two more proteins which are attached to actin. Those proteins are tropomyosin. You can see this green line is representing you the tropomyosin. This is a fibrous protein. This is not globular protein, while actin is a globular protein. Now, tropomyosin is actually covering the heads of the actin. You can see in this way the heads of the actin are covered. Okay, now the what is the function of this tropomyosin if the heads are covered in this way? Actually, if heads are covered by this tropomyosin, then these heads cannot join to these heads. These heads are present on the thick filament. So basically, during the contraction of the muscle, these the heads of the thick filament are joined to the heads of the thin filament. But during the normal situations, the tropomyosin will cover the heads of this actin so that the actin heads cannot bind to the globular heads of the myosin. Okay, now there is another protein which is known as the troponin. You can see this red region is showing you the troponin protein. This troponin protein will bind to calcium, and calcium is the stimulus for this complete cycle in which the muscle is contracting, and whenever the calcium is binded to the troponin, now there will be the binding of the heads of the myosin to the heads of the actin. So but in the normal situations the heads are covered by the tropomyosin. Okay, so this is the structure of the thin filament. Actin is present, tropomyosin is present, which is covering the head of the actin, and another protein which is known as a troponin, and this is the calcium binding protein. Now come to the next filament, which is the thick filament. So this is the thick filament. Thick filament has the two part, the fibrous part and it has the globular heads. Now the function of the globular head is very special, that the head has the two regions. You can see the globular heads have the two regions. So the one of its region will bind to the actin, and while its another part has ATPase enzyme, and ATPase enzyme will definitely hydrolyze the ATP so that the energy needs can be fulfilled and there could be the contraction of muscle. So this is all about the structure of myofibril. Now we have further diagrams you can see in your course book. So this is one complete muscle fiber. Now when we see the structure, then you can find out that it has a sarcomere, and as I have mentioned, in the sarcomere the two light regions and one dark region is present, and sarcolemma is present in the muscle fiber. Sarcolemma is what? The cell membrane. Cell membrane of the muscle fiber is known as sarcolemma. Now in this figure you can see this is the external structure, and in this figure you can see that This region is the green color. This is the sarcolemma. Now, sarcolemma has some invaginations in this way. You can see the sarcolemma has invaginations in this way. These are the invaginations of the sarcolemma. Now, these invaginations are actually the forming the T tubule. The T tubule is made by the invagination of the sarcolemma. And T-tubule is a triad structure because on both of its sides it has the sarcoplasmic reticulum. This is showing you the sarcoplasmic reticulum. So sarcoplasmic reticulum is present on both of its sides. So this is making the T-tubule system. This is the T-tubule in which the sarcolemma has some invagination, and on both of its sides it has the sarcoplasmic reticulum. So this is the T-tubule. And what is the function of T-tubule? The function is when an action potential arrived at sarcolemma, then the sarcolemma depolarized first. Now the action potential is traveling downward through the T-tubule. So that's why there is an invagination just like T-tubule. Now you can see furthermore, these figures are also in your course book. And this is the structure of the myofilament. Now, furthermore, when we see, this is the internal structure, and in the internal structure, you can see there are different regions, light regions and dark regions. So these are the dark regions and these are the light regions. But there are some regions like this. These regions have both type of bands, means dark band, and the parallel to this dark band, there is the light band as well. So these regions are also found. So the region where the dark band dark band is present, and also it has the region where both type of bands are meeting, means in the parallel way both type of bands are present. So that region is known as A band. This is A band. A band is uh, complete the dark band. What is the H band? H band is the region which is only representing you the dark region. So this is the H band. And what is I band? I band is completely showing you the region where light band is present. So these are the three bands which are present on the myofibril: A band, I band, and H band. Okay, now you can see there are some lines which are present. That is the M line and Z line. M line is actually holding the thick bands. These are holding the myosin proteins, myosin filaments, or thick bands. And what about the Z line? Z line is holding the thin filaments. You can see the thin filaments are held by the Z line. And what is the one complete sarcomere? You can see two Z lines. Z line is holding what? The light bands. And actually in the sarcomere, the two light regions and one dark region is present. So you can see that the two Z lines and one M line is present in one complete sarcomere. So this is all about the structure of muscle.